All right. Okay, maybe I wait a minute until everyone found a spot. All right, good. So thanks for the invitation, Philippe, for talking in this session. I will be talking about this kind of how we can use genetics to identify drugs in a much more basic research setting. So I'm a basic researcher. And one question that I find actually very interesting and very fascinating, and Nicolina has already touched a little bit upon this, is to understand what makes each individual, or you can also say each patient, unique. Rather than with unique, of course, you mean, when you look at everyone, everyone has a different face, we have different kind of phenotypes that can be the height. But also we have seen in the recent pandemic, for example, that other features such as age are actually quite important for determining kind of the phenotypic outcome of something like, for example, an infection. And what I find fascinating, what we are using a lot in our research is trying to use this variation to understand basic biology, and with that also disease mechanisms. Now, when we talk about variation across individuals, we really have to consider two sources. We've heard a lot about the genetic source from the previous talk from Nicolina, and I think the genetics field, the field of human genetics in particular, has made tremendous progress in the past 20 years or so. And we have now been able to link over 200,000 of these single nucleotide polymorphisms to complex traits and diseases. On the other hand, we have, of course, all these extrinsic factors such as infections, mutations that can happen, our nutrition, the microenvironment, and so on, that all have an influence on the phenotype as well. Now, what are the big challenges in those two aspects? I think one of the big challenges with the GWAS, with the genome-wide association studies, is that they all fall in these non-coding regulatory regions, most likely regulatory regions. And we really don't understand very well what they're doing. And the other effect is the other side, from the extrinsic features. I think that the challenge is even more complex because we don't even know what's associated. So now, if we consider that the cell is really the basic unit of an organism, and that's where the phenotype kind of eventually emerges, we can ask ourselves, what is the intrinsic effect of the cell state of these two factors, right? And so we really need to understand these molecular correlates of phenotypic variation. Now, why does this matter? I'm just going to go here in a very idealistic world. If we have a precision medicine in an idealistic world, if somebody would fall ill, we would actually get the genetic profile of that person, understand kind of the lifetime risk or the propensity of that person to become ill with a certain disease. We would get all the extrinsic factors that the person has been exposed to, calculate some kind of cumulative risk, and get a molecular understanding of the disease, which would then help us to get 100% fitting therapy. Now, of course, we all know that this is not possible because we will never get there. At least I don't believe we will get in my lifetime ever to get there. So what we're doing instead is we're using biomarkers to understand risk stratifications and try to make therapy decisions or design drugs. And in my view, biomarkers in a way are kind of the molecular correlates of phenotypic variation, right? And if we now think about what is driving these molecular correlates of phenotypic variation, and we go again back to these genetic and extrinsic factors, when we think about the genetics, I think we can say the coding variants may have a direct effect on these molecular correlates. Right? But when we look at the non-coding variants, essentially we already see that they may act through gene regulation. Similarly, the extrinsic factors, they may have a direct effect on these biomarkers or molecular correlates of phenotypic variation through signaling and metabolism, but they also may have a signaling effect that ends up at a transcription factor that again affects gene regulation. And then we have all these extrinsic factors that affect epigenetic features, which again may affect gene regulation. So I think you see where I'm going with this. I think gene regulation is really an important process to understand if we want to understand the molecular correlates of phenotypic variation, and with that understand kind of the mechanisms that may drive disease. So what is gene regulation? I think I just want to get you a very basic understanding of how I view gene regulation. Essentially, we have this nuclear compartment where we organize the chromatin. We have this three-dimensional loops where promoters link to enhancers. And if we now just do this simple thought experiment and we think about what may change the expression of a certain gene, which is kind of the basic layer of phenotypic variation, of course, the absence of a transcription factor may repress the gene, so the gene may not be active because of signaling, because of a transgenetic effect, such as a mutation in transcription factor, or because of the cell type or cell state. Then, of course, we can also have cisgenetic variants that may affect transcription factor binding through a single nucleotide polymorphism, so transcription factor cannot bind there. Again, we would lose transcription. And finally, we can also have chromatin accessibility that prevents a transcription factor from binding to some epigenetic mechanism. So this would be an epigenetic mechanism, or kind of, again, reflecting maybe cell type or cell state. So cell type and cell state are really important contributors for kind of understanding what may drive differences in gene expression. And this really brings me to our vision that we have in a group, which is to use variation across individuals and integrate these multiple layers of genetics and epigenetics to understand at the molecular level the disease mechanisms. So essentially we do molecular profiling. We try to build these gene regulatory networks that connect the signaling component through the transcription factor to the regulatory elements and then the target genes. And we then can integrate these different ways of extrinsic signal genetics and epigenetics. And ideally, we can then build these networks in a cell type specific way to understand the perturbations that may happen through a specific extrinsic or intrinsic perturbation in different cell types. So we use variation across individuals, and I just want to briefly motivate why we're using variation across individuals as a way to kind of generate these models, because of course when you generate a network, it's always important to understand what are the edges of your network actually representing. In our case, it's really kind of variation, covariation across individuals. And this comes from a really basic research study that we have done a couple of years ago that was driven by Olga Sigolova, who wanted to understand why are some genes very tightly regulated and essentially don't vary at all across individuals, whereas other genes show a huge variation if you check across individuals. And what she was doing is she was collecting all kinds of genomically encoded regulatory features and then built machine learning models to predict variation across individuals. And we did that in two systems. One was in a very controlled system of Drosophila embryos, genetically diverse, 100 at a time. And the other one was a really messy system of postmortem samples from the GTEx consortium of human tissues. So Olga built a random forest model to try to predict variation across individuals in those two settings. And what was quite intriguing to me is that we could actually predict variation across individuals just based on these genomically encoded features with an R-square of 1.2 with a random forest model. Very simple. And this was the same for Drosophila and human. Now, what was even more fascinating, if we now took the database of differentially expressed genes, so not variation across individuals, but genes that actually respond to any kind of perturbation, we found that the same model would actually predict very well whether a gene would be differentially expressed or not across any random condition. I think it was a total of 700 conditions that they collected actually in this paper there. And what we found is essentially the most important features that predict variation across individuals or variation in response to a perturbation is essentially the regulatory complexity. So that is very much in line with the concept of buffering, where if you have a complex promoter, you could the orange gene would be activated if you have the green transcription factor, if you have the blue, or if you have the red. Whereas the blue gene with a very simple promoter really requires this one transcription factor to bind. So as soon as that transcription factor is not there, the gene would not be active. So this is essentially what we found as the most obvious kind of explanation of what explains variation across individuals. And I think overall what this study tells me is that variation across individuals may recapitulate essentially a gene's ability to integrate, respond to external signals. And that's why we use a lot in our research this variation across individuals as a way to integrate also the extrinsic signals. All right, so let's go to gene regulatory networks. There has been lots of approaches, and I'm not going to spend much time on this. Essentially, what we wanted to do is not going straight from the transcription factors to the target genes, but in our case, we really wanted to include the enhancers. And I'm sorry for the mess up there. That's a PowerPoint issue. Essentially, we wanted to understand we wanted to integrate the enhancers in our models. All right, so what do? We do a very simple model where we essentially split these task in the two, and we, on the one side, use covariation across individuals to link transcription factors to enhancers or to regulatory elements, and then again, use a very simple model, again, to use covariation across individuals to link the enhancer to the target gene. So it's a bipartite model that links transcription factors to enhancers to target genes. By the time we have done this was the first one. Now there's other models around out there that do kind of similar things. And there was a poster yesterday, so if you're interested more on how we can actually apply this also to single cells, Christian Arnold has a, by now, virtual poster about this. But what I want to go next is to actually, so we have spent a lot of time in actually understanding whether these networks are biologically meaningful, right? Because anyone can make kind of a network, you know, and you need to be able to understand whether it's useful. And this is now, we have done extensively benchmarked our network with that we derived from naive macrophages. And I also want to point out here that this is really a difficult task because there's no gold standard at the moment for validating gene regulatory network. And there's actually a joint effort ongoing also with Julio's lab. Pau is the one driving it, where we are trying to put together a standard for evaluating gene regulatory networks. But I'm not going to talk about this work here. I'm just going to point out a couple of challenges when you try to evaluate gene regulatory networks. So you can, of course, look for molecular ground truths and ChIP-seq and eQTLs. And this is what we have done for our network. And essentially here you can validate the individual links, right? You can see how well can we actually recapitulate ChIP-seq data from our transcription factor to regulatory peak links. And the challenge here is that there's only a few data sets available for cell type specific transcription factors. Then I think the more kind of established one is the eQTL links, where you can try to link your enhancers to genes, and you can validate that with expression QTLs. And here again, we showed very nicely that we can actually do that with our networks. You can perturb the TF edges, but this gives you a lot of very indirect effects. That's my second point. And the third point, I think, is quite important, actually, also as a take-home message, because, of course, you can now perturb these enhancers with CRISPR-I. And that's just showing you here a figure that actually has on the one axis has the expression variation that I've shown you before, in the first part, you know, about that we kind of that we look at variation across individuals versus the number of CRISPR-I perturbations that perturb a specific gene. And what you can essentially see, the more variable a gene is across individuals, the more likely it's going to be perturbed by any CRISPR-I perturbation. Right? So again, I think you have this intrinsic genetic gene bias when you want to kind of evaluate your enhancers with CRISPR-I. Anyway, then we have a lot of synthetic and simulated data. I'm not going to go into detail, but what I'm going to go here is that we maybe can use something more to go a bit beyond description and use the networks to actually predict something. And what they should be able to predict, of course, is gene expression, right? Because there's gene regulatory networks. So what we have defined for us as a way of identifying useful gene regulatory networks is that we want them to be able to predict differential expression. I'm showing you here the example again of macrophages that were stimulated with Salmonella. And we want to predict essentially based just on a network, how well can we recapitulate differential expression data. You can see here the predicted log twofold change versus the true log twofold change. And we get an R-square of about 0.25. Now, obviously, we don't want the random network to be able to predict this, so we shuffle our edges and we get essentially 0 prediction. So that's very good. We are better than random. But in addition to being better than random, of course, we also want to be better than a cell type generic network because we think gene regulatory networks are very cell type specific. So indeed, when we now build specific gene regulatory networks across different cell types. What you can see here on the y-axis are the different cell types for which we have specific perturbations. And on the x-axis are the different gene regulatory networks that we've built. And you see that the networks essentially can only predict well in the cell type for which they have been built. So this is always, importantly, always the data that we use for the stimulation is independent of the data that we use for generating the network. All right, so now I think we have a case for really saying that we need these cell type specific regulatory networks. What can we learn from them? And I think it's very important that we can now use them to understand disease variants a little bit better, right? So we can now go to these fine map variants. We can look at what are the transcription factors that are regulating these variants, and we can now go in a genome browser, and you can see that often it is not necessarily the closest gene that is regulated by a specific SNP. Here is one example for a SNP associated with systemic lupus erythematosus. And of course, now we need to follow up these experiments, these things again with experiments. How can we now integrate non-genetic perturbations? And I think this is really kind of the idea that we have when we now have our gene regulatory network that we trust, and we can now predict a specific set of differential expression. We can identify now the important transcription factors. And that is now the key, how we can now integrate the differential, the kind of the extrinsic signal. And again, a poster application to single cells can be found online by Christian Arnold. And Ariane has been giving a talk about this. So what I mean with that, so we can now again take our macrophage network. We have these different perturbations, different stimulations of the different infections essentially, and we find each infection has a different transcription factor that comes up as being important. We can then validate some of these, and FKB comes up on interferon gamma stimulation, and we find that breast cancer-associated macrophages are driven by very specific transcription factors such as TRD1, and the infection, we identify a new transcription factor that may be driving infections, which is PRA, and which actually validated with phosphoproteomics data. It really does get activated upon macrophage stimulation. So this is now this is how we can now get the insights in the non-genetic disease mechanisms, if we can now integrate the genetic variants from a disease and the environmental stimulations, for example, for infection or for disease-specific modifications. And so I have, how many minutes I've left? 4 minutes left, okay. Okay, good. So maybe I just end with this kind of final summary that we have. So essentially, we used the insights that we got from our cell type specific networks is that really the important thing between cell types is that transcription factors can have very distinct targets across cell types. And another thing that is very important to remember is that enhancers can actually define the functional potential of a cell by priming. And what I mean with that is that you can have some cells that have an active enhancers, and if the signal comes, the gene actually gets expressed, whereas in an inactive cell, you don't get this signal expressed. And I think I actually stop here and maybe go to my last slide. Yeah, so essentially what I wanted to bring across here is that we have a framework for integrating the genetic and the environmental factors, and I've shown you a couple of examples of how we can do that. And these are the acknowledgements. Thank you.
Thank you. Questions?
Thank you so much for the great talk. We know that there is a lot of overlap on enhancer activity that have been shown in between individuals. And this is actually helping to reduce the effect of environmental or even genetic defects that can exist. Do you have any evidence of some of those SNPs that do not actually, that the prediction don't fit with the observations if they are enriched on those in overlapping enhancers? Enhancer with overlapping activity.
No, I haven't looked at that, but that's a good point. Yeah, no, I haven't. We haven't looked at enhancers of overlapping activity.
Thank you.
Yeah.
Hi, thank you for the nice talk. I was wondering, now that we have cell type specific gene regulatory networks, and we also know that cells communicate with each other, how do you envision to integrate the data between cell-cell communication and gene regulatory networks?
Yeah, this is a great question. This is exactly what we're trying to do in the future. I think many people are working on this, but one way is, of course, to kind of understand the connection between a receptor and the transcription factor without going through all the signaling in between, right? You can essentially try to do that from single-cell RNA-seq data as well. Yes, but I think this is the future, indeed.
Okay, last question.
Thank you. I'm new to it. I haven't got any experience on multiomics, but I just wonder if these regulatory network is sort of complete, or do we have almost finished constructing these networks, or they're still like work in progress now?
I think it's work in progress. I mean, in a way, you need a new gene regulatory network almost for, I mean, depending on your question, right? I mean, we have now some for specific cell types, but I would say depending on the cell type, you would need to generate your gene regulatory network. Yes.